Hey everyone, and welcome back to the Biochemistry YouTube channel. Today, we will talk about glycolysis, which literally translates to splitting sugars. In the process of releasing energies in glycolysis, a 6-carbon sugar known as glucose is split into 2 molecules of a 3-carbon sugar called pyruvate. This ends up yielding 2 ATP molecules containing free energy, 2 pyruvate molecules, 2 NADH, and 2 molecules of water. Because glycolysis does not require oxygen, it can be aerobic or using oxygen or anaerobic without oxygen. Glycolysis is a somewhat inefficient process. This is because much of the cellular energy remains in 2 molecules of pyruvate that are created, which end up being more productive in this entire process later on. In the presence of oxygen, glycolysis is the first stage of cellular respiration. In the absence of oxygen, glycolysis allows cells to make small amounts of ATP through a process called fermentation. Let's dive straight into glycolysis. The first step of glycolysis is the conversion of D-glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is called hexokinase. Here, the glucose ring is phosphorylated. Phosphorylation is the process of adding a phosphate group to a molecule derived from ATP. As a result, at this point in glycolysis, 1 molecule of ATP has been consumed. Hexokinase catalyzes the phosphorylation, as I just mentioned. Magnesium is seen here because it shields the negative charges from the phosphate groups on the ATP molecule. The result of this phosphorylation is glucose 6-phosphate. The second reaction of glycolysis is the rearrangement of glucose 6-phosphate into fructose 6-phosphate by glucose phosphate isomerase, also known as phosphoglucose or phosphohexose isomerase. The second step involves the conversion of glucose 6-phosphate into fructose 6-phosphate. This reaction occurs with the help of this enzyme. As the name of this enzyme suggests, this is just an isomerization reaction, meaning it involves the rearrangement of the carbon-oxygen bond to transform the 6-membered ring into a 5-membered ring. Phosphofructokinase, with magnesium as a cofactor, changes fructose 6-phosphate into fructose 1,6-bisphosphate in step 3. Similar to the reaction that occurs in step 1, a second molecule of ATP provides the phosphate group that is then added on to fructose 6-phosphate. The enzyme, like I mentioned before, is phosphofructokinase. Same like in step 1, a magnesium atom is involved to help shield negative charges. But I do want to make a point here. Let's stop and see something. This is the rate-limiting step, right? This enzyme, phosphofructokinase 1, is super important. This is because it has a sister enzyme known as phosphofructokinase 2. This is why it is known as bifunctional. This bifunctional enzyme is called that because of its notable structure. Though it is located on one protein homodimer, its 2 domains act independently as functioning enzymes. One of them serves as a kinase domain, while the other serves as a phosphatase domain. This has much to do with the regulation and hormonal regulation of insulin and glucagon, 2 hormones used to regulate hunger. Anyways, let's get back to phosphofructokinase. Again, remember, it uses ATP to phosphorylate fructose 6-phosphate into fructose 1,6-bisphosphate. This is also very important because it is the rate-limiting step. The enzyme aldolase splits fructose 1,6-bisphosphate into 2 sugars that are isomers of one another, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. As suggested, because we have 2 isomers, they both need to turn into one of the forms, in which in step 5, trisubphosphate isomerase rapidly interconverts DHAP into G3P. This is done because G3P is the only molecule that can continue in the glycolytic pathway. As a result, all DHAP molecules are turned into G3P. At this point in the glycolytic pathway, we have 2 3-carbon molecules, but we're not at pyruvate yet. Glyceraldehyde 3-phosphate dehydrogenase dehydrogenates and adds an inorganic phosphate to G3P, producing 1,3-bisphosphoglycerate. In this step, 2 main steps take place. The first one is that G3P is oxidized by the coenzyme NAD. Then, the molecule is phosphorylated by the addition of a free phosphate group. Phosphoglycerate kinase transfers a phosphate group from 1,3-bisphosphoglycerate to ATP to form ATP and 3-phosphoglycerate. In this step, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate by the enzyme phosphoglycerate kinase. This reaction involves the loss of a phosphate group from the starting material. The phosphate is transferred to a molecule of ADP that yields our first molecule of ATP. Since we actually have 2 molecules of 1,3-bisphosphoglycerate, because remember, there were 2 3-carbon products from stage 1 of glycolysis, we actually synthesized 2 molecules of ATP at this step. With this synthesis of ATP, we have canceled the first 2 molecules of ATP that we used, leaving us with a net 0 ATP at this stage in glycolysis. Again, we see that an atom of magnesium is involved to shield the negative charges on the phosphate groups of ATP. In step 8, phosphoglycerate mutase relocates the phosphate from 3-phosphoglycerate to the second carbon to form 2-phosphoglycerate. This is just a simple rearrangement of position of the phosphate group using a mutase enzyme. Step 9, the enzyme enolase removes a molecule of water from 2-phosphoglycerate to form phosphoenolpyruvic acid, also known as PEP. The enolase works by removing a water group, or dehydrating, the 2-phosphoglycerate. Our last step uses the enzyme pyruvate kinase, which transfers the phosphate from phosphoenolpyruvate to ADP to form pyruvic acid and ADP, our final product. One more thing to note is that if we are in an anaerobic condition, meaning we are not using oxygen, an initial step takes place to form lactate. When animal tissues cannot be supplied with sufficient oxygen to support aerobic oxidation of the pyruvate and NADH produced in glycolysis, NAD+ is regenerated from NADH by the reduction of pyruvate to lactate by lactate dehydrogenase. But that's all I've got for you today. Make sure to watch our next video about gluconeogenesis. Good luck on exam 3!